Good morning, Year 9. It's Miss Rasmussen here with your Week 3 lesson. We're going to look at osmosis today. So we will start off with our questions. So question 1, what does active transport need to happen? Question 2, how do particles move in diffusion? Question 3, how do you calculate magnification? Question 4, which organelle contains genetic material? And question 5, name 3 organelles found in plant and animal cells. So please pause the video, answer the questions, and then come back to check your answers. Okay, Year 9, welcome back. We're going to check our answers. So in order for active transport to happen, you need to have energy. So remember, active transport is against the concentration gradient, so we need energy. How do particles move in diffusion? They move from an area of high concentration to an area of lower concentration, so from high to low. How do you calculate magnification? So for this one, you need to rearrange the equation. Magnification is image size divided by actual size. Question 4, which organelle contains genetic material? That is the nucleus. And then finally, name 3 organelles found in plant and animal cells. So this is any of the shared organelles. And you can have any 3 from nucleus, mitochondria, cell membrane, cytoplasm and ribosomes. So see how many you've got. Hopefully you've got them all right. These are all from your facts that you need to be learning from your fact book. Okay, so what we're looking at today is osmosis. On core, this is your only lesson for the week. If you're on triple, this is your first of 2 lessons. And what we need to be able to do is define osmosis. So osmosis is the movement of water from a dilute solution to a concentrated solution through a partially permeable membrane. You need to be able to talk about how cells can change mass in osmosis, and that is from water entering or leaving the cell. You need to say what will happen to a cell put in a concentrated or strong sugar solution, and that will shrivel up and lose mass. And then you need to be able to do the reverse, so a cell put in a weak sugar solution will swell up due to water entering the cell. Okay, so start with our definition. Osmosis. What osmosis is the movement of water from a dilute solution to a concentrated solution through a partially permeable membrane. So if we just break that down, movement of water. So when we are talking about osmosis, we are only talking about water. The other particles are too large and they're not able to move. From dilute to concentrated, so what we're talking about with this is dilute, where there are lots and lots of water particles, to concentrated, where there are not many water particles. Again, think back to your squash. Dilute, really, really pale, lots of water, not much squash. And then concentrated, much, much darker, because there's a lot more squash and therefore less water. And then finally, a partially permeable membrane. For example, the cell membrane that only lets certain molecules pass through. So let's just have a look at this idea of how the water is moving in a little bit more detail. And here's an example. So if you start with your dilute solution, so here we've got our dilute solution, and on this side we've got our concentrated solution. This line here is our partially permeable membrane. So the way that water moves in osmosis is from a dilute to a concentrated. So the water is going to pass through this membrane. And then what happens is you then get the 2 solutions. They've got the same concentration, but you can see that the volume of this one has gone down and the volume of that one has gone up. And that's because the water has to move to make these concentrations the same. These molecules here, which could be sugar, could be salt, can't get through this membrane. Now, it's a really funky experiment that you can do with eggs, and what I would like you to do is watch the video. It is in the comments below, so if you can go and watch that video and have a look at what happens to the eggs in osmosis, and then come back when you're done. Okay guys, so welcome back. You should have watched that video on the eggs. Next task, what I would like you to do is just do your own explanation. So I'd like you to pause the video again, draw out this diagram, and then use it to explain what is happening in osmosis. So use it to explain osmosis. So you can link to what happened with the eggs. So this example here would be when your egg was shriveling up because the water was leaving. Okay, so next thing, just to apply our understanding, what I would like you to do is just on each beaker, have a think about it, work out which way the water will move and use an arrow. So in beaker 1, inside the cell, you've got 55% water and 45% solute. And then in the beaker, it's 45% water and 55% solute. So work out in each one, which way will the water move? Into the cell, out of the cell, or will it be the same? Okay, so here are your answers, folks. What you've got is in beaker 1, it's going to be moving out of the cell because it is more concentrated outside. There is more water inside. Okay, then in beaker 2, the water is going to be moving into the cell because inside this cell, it is much more concentrated. So it's a 75% solute compared to 40% solute outside. And then finally, beaker 3, what's going to happen here is the water is going to move in and move out. So the overall movement will be 0. And that is because the concentrations inside and outside of the cell are the same. Okay, so now we're going to look at another example of osmosis in action and changes in mass. Now, just to let you know, no gummy bears were harmed in the making of this image. And it is also something that you can try at home. Just make sure people at home are happy with you doing this and that you're not eating some Haribo that have been saved for a special occasion. So what we've got is we've got our original gummy bears. So this is what they would normally look like. And then they were placed in different solutions for 24 hours. So here you've got your gummy bears that were put in salt water and they look really, really shriveled and sorry for themselves. Sugar water, you can see they've puffed up a little bit. They're relatively big. They're definitely bigger than the original. And then finally, just regular tap water. They have got massive. You've got really big, giant gummy bears. And this is all to do with osmosis. So if the water will enter the cell, the mass is going to increase. And this is what's happened here. So the sugar water, the concentration of the sugar inside would be higher than the water, so the water will move in. But in regular water, the concentration gradient is even bigger, so more water moves in. So the mass on these two has increased. In the salt water, because the concentration of the solution outside was higher than inside, all the water has left. So if water leaves the cell, then the mass will decrease. So what we need to know, and this is in our fact book, is that cells change mass in osmosis, and this comes from water entering or leaving the cell. So what I'd like you guys to do is do a little bit of an explanation. So I'd like you to pause the video again, and then whilst you've got it paused, I would like you to choose 2 of the gummy bear images from salt water, sugar water, regular water, and explain using osmosis what has happened to them. Some keywords that I'd like to see, and please make sure that you underline these as you go through. Osmosis, water, movement, concentrated, dilute, and mass. So make sure you are putting these keywords in. So you're choosing 2 of the gummy bears and explaining what is happening using the idea of osmosis. Okay guys, so welcome back. Last thing we're going to do is just have a quick look at an exam question. They love asking ones about this, about how these masses are going to change. So some students set up the equipment below to investigate osmosis. We've got a capillary tube. This is just a thin glass tube. Beaker, we've got water in our beaker. And then this is visquing tubing with concentrated salt solution. Now visquing tubing will act as a semi or partially permeable membrane. So your first question is, what is osmosis? Then your second question is, what will happen to the water level during the investigation because of osmosis? And then part 2 of that is, use your knowledge to explain why this happens. So please pause the video, have a go at these questions and come back to mark them when you're ready. Okay guys, so welcome back. First of all, what is osmosis? We can use our definition that we've learnt here. It is worth 3 marks and this will give us the 3 marks. So it is the movement of water, that's our first mark, from a dilute solution to a concentrated solution. That's going to give us our second mark, through a partially permeable membrane, and that's our final mark. Then what will happen to the water level in the capillary tube during the investigation because of osmosis? So this just wants you to say factual statement. So what's going to happen is, because the water is going to be going in, it's going to be moving up. And that leads us on to our explanation. So thinking back to osmosis, inside here is going to be your concentrated solution and here is going to be your dilute solution. So the water is going to move into the tubing and that is then going to push that water level up the capillary tube. Okay, so what you need to know from osmosis, these are in your fact books as well, so please make sure that you are learning them. Any problems, please contact a member of the science staff. Osmosis is the movement of water from a dilute solution to a concentrated solution through a partially permeable membrane. Cells change mass in osmosis from water entering or leaving the cell. A cell put in a very concentrated or strong sugar solution will shrivel up and lose mass. A cell put in a weak sugar solution will swell up due to water entering the cell. Okay guys, so that's it for your osmosis. I will see you on the next video, which is going to be about stem cells. So take care, look after yourselves and keep washing those hands.